I talk a lot about other people's science on my page, but I'm also a scientist, and I just published a paper on my birthday, and I want to tell you about it. So in my PhD, I studied the neurobiology of autism, and I found that mice with an autism-linked gene mutation actually had too much brain activity and a part of the brain that controls social interactions. This may sound like a good thing, but it's actually not ideal, and it made these mice less social. Then I found that if we turned on this one protein, we could essentially build more red lights in the brain, and that would reduce the brain activity in that area. And doing that actually made the mice more social. But the problem is, there's really no easy way to turn on that protein. It's supposed to turn on when the cells are active, so the only way to turn it on would be to activate the cells. But the problem is, the cells are already too active. So how the heck do we approach this? Well, in this paper, which was just published, I found that we could target a different protein which controls the protein that we want. And doing that activated the same process in the brain and also made the mice more social. Now, the good news is, this discovery makes this way more feasible as a treatment in humans. But the bad news is, mice are not humans, and these types of findings never seem to translate to humans. When I say that, I mean there are 0 available treatments for the social symptoms of autism. So this is hopefully a step in the right direction for those who may want a treatment but have no options. An important final note, I stand with the ASD community, and I absolutely do not view autism as something that should be cured. I simply and genuinely am just doing my best to help others through science. So thank you so much for watching and for your interest.